Hello, everyone, and thank you for joining us. My name is Ben Pastro, and I am the COO of Clovertex. While Clovertex is based in Boston, we're grateful to have over 100 participants from across the US and Europe. We are experts from large pharma companies, biotechs, and academia, and we range from students to folks who are well into our careers. And despite this variety, I think we can all agree that we are a community. We are a community of people who work in the world of cryo-EM and who are all passionate about making the world healthier through this work. We're also a community of people striving to help one another. And that is really at the heart of this webinar, helping one another, and through that, further strengthening this community. So as my friend Mazdak from CryoDuck wrote in the invite, we will be discussing the unique aspects of processing cryo-EM data in industrial settings. We hope to make this a lively exchange amongst our panelists and our attendees, so I encourage everyone to ask questions. You can use the Q&A button on the bottom of your screen. And any questions that we don't get to, we will answer offline. Mazdak will be helping to facilitate this. I mentioned that I'm with Clovertex. We are an IT partner for life sciences, specializing in helping R&D teams accelerate drug discovery. We have deep scientific domain and IT expertise, and one of our superpowers is significantly improving the throughput and the quality of cryo-EM data processing with cloud infrastructure tailored to our customers' needs. Our panelists today, and we're very grateful to have them with us, is Katie Meze, who's with Sanofi, Roberto Valverde with Renaissance Bio, and Simone Cavadini with the Friedrich Miescher Institute for Biomedical Research, FMI. So welcome, and I'd like to ask you to introduce yourselves, and I'd like to do that through a question. Cryo-EM is widely used in both academia and industry, but the environment, priorities, and workflows can be quite different between the two. So for our panelists, can each of you introduce yourselves and share your journey into industry and what you see as the key differences in working with cryo-EM in industry versus academia? Katie, will you kick us off?
Sure. Thanks so much, Ben, and thank you so much, Mazdak and the whole Clovertex team for this opportunity to have a really interesting discussion. I actually I'm not from the US, as you might have guessed. I come from Europe. I grew up in Slovenia, and I did my undergraduate in the UK in chemistry, and that was really where I fell in love with structural biology. I had some amazing mentors there, and so they helped me sort of find my interests. And sort of since then, I've been going west, and I ended up in Cold Spring Harbor for my PhD, working with Limor Joshua Tor. Really fell in love with cryo-EM there, and the opportunities and the insights that cryo-EM can give us into the way that biological machines function. After that, I did a short postdoc with Melanie Ohi, who is an expert in cryo-EM. That was a really fun experience, and she was a fantastic mentor who also really helped me then make the transition over to industry. So I guess the big differences that I've noticed sort of going from academia into industry, I mean, there are many, from, the way that you do your science to sort of the priorities and the timeframes that you're operating in. But I think in terms of cryo-EM, some of the major things I was quite apprehensive about was first that a lot of industry cryo-EM facilities don't have a cryo-EM manager. So that was something I was a little bit unsure about how it would work. And then the second thing I think I really noticed was that also the cryo-EM data processing infrastructure, whereas, in academia, it's generally quite bespoke for the group. So you have specific sort of infrastructure, either, well, mostly on-prem, but also some cloud computing. Whereas in industry, you have to fit that into the way that the larger company operates. And so that takes a little bit more working with IT and being sure that you can communicate your needs as a cryo-EM scientist for them as well. So there's many other sort of small differences, but maybe I'll just leave it there for now. Let the others introduce themselves.
Thank you, Katie. I guess I'll go next and just want to also thank the Clovertex team and Mazdak for this kind invite. Just by way of introduction, my name is Roberto. I did my PhD at Yale University in the laboratory of Lynn Regan, where I got to do some cool protein engineering and use crystallography. From there, I think I'm from a generation of crystallographers that sort of like bigger was better. I wanted to go on to like harder, more complicated structural biology problems. So I did a postdoc in Steve Harrison's lab. And at that time, around the time that my postdoc project was finishing up, the new detectors came online. So I actually never learned Cryo-EM in academia. All my Cryo-EM experience and training comes from industry. And so I often describe that as learning to fly the plane as you're building it. So I think in terms of answering the first question, I can talk generally about structural biology. I think just the general difference between academia and postdoc. I think in academia, again, this dates me. I come from a generation of structural biologists where we were all trying to work to get the first in class structure. So seeing that, protein complex for the first time. And so whether a crystal system or maybe even a cryo-EM system gave 3.5 angstrom resolution or 2 angstrom resolution, that was good enough to model it and publish a paper. What's very different about doing that sort of work in industry is that it has to become, your structural biology system has to be very robust, and you have to be able to reproduce it in such a way that you can sort of, if you're a small molecule company, use that system or iterate on that system to drive SAR. And so really thinking about making structural biology robust, reproducible, and to Katie's point, making it all work within the existing infrastructure is, I thought that was one of the key difference for me, at least.
I guess continue. Welcome everybody to this webinar. Thanks Ben, Clovertex, and Mazdak for organizing this. Very happy to share my experience today with you, and happy to answer your questions when you have. So my name is Simone. I'm Swiss, and I study chemistry in Lausanne, and then I moved as a postdoc at the Friedrich Miescher Institute, where basically I learn how to purify proteins and start imaging these proteins with a microscope. So at the time, we were still in the blobology times. So I have many colleagues at the time that were making fun of my work. But I think now, as you all experience the Cryo-EM resolution revolution, this kind of payback. And I'm happy to be in this field. Yeah, so the FMI is a research institute embedded within Novartis. So we kind of represent the academic part of Novartis. So we are basically more free to explore project, but being embedded with Novartis, of course, there is the benefit of sharing technologies and so on. So I guess leaving this interface, I can see many differences, but also many similarities between academia and industry. And maybe the environment, as was mentioned briefly before, is a bit different. So you, as academic, you mainly interact with PhD, postdoc group leaders, while of course industry, you interact with other type of scientists, like chemists, biologists, and so on. What I also maybe experience, and we can elaborate a bit during discussions, is the fact that in academia we have, I would say, quite different type of samples. I can see that the industry usually the target are very well defined. So you tend to work with targets of proteins that have been purifying since decades sometimes. So the proteins are very well structured, suitable for drug discovery, of course. While in academia, very often we handle with protein complexes that needs to be reconstituting from scratch for the first time. So there are, this type of challenges on the sample level, which are pretty obvious when you compare academia and industry, I think. But yeah, maybe we can continue.
Thank you all for introducing yourselves. Simone, I'm curious, do you see the model that you operate in at FMI? Do you see that being a model that will continue to expand over time? Will there be more examples where folks like yourself are working at that intersection of academia and industry?
Yes, definitely. I think this really paid off for Novartis. When we so the FMI, we are doing EM before Novartis, right? So this was, as I said before, in the globology time. But we had the technology now, right, ready to be used, also exploited by industry. So we were growing our experience, and Novartis, as soon as the technology was ready to go to high resolution, basically was not a click, but much easier for them to jump on the technology based on our experience. And I see this coming also in other fields. So I think this collaboration is definitely something that is worth keeping and investing in the future.
Great, thank you. So Katie, in your intro, you mentioned mentorship, and mentorship is often emphasized in academic training. I'm curious, in your journey, what does it look like to you in the industry setting, and how have you navigated mentorship, being a mentee as well as being a mentor?
Yeah, it's a great question. I think that's something that a lot of people who are considering going into industry think about, especially because I think a lot of us really enjoy mentoring others, and it's really kind of the key part of academia. So I think, as you mentioned, we can think of it in both senses. Sort of when you're coming in, trying to enter into industry, I received a ton of mentorship from people that I connected with. These were, second or third colleagues, maybe, that helped sort of tell me about what the differences were, how I should best apply to industry, what to look out for, and so on. And that can be pretty daunting, I think, especially if you don't not just have connections from friends in industry. And so I'd like to highlight one of our groups that we've put together to especially, I think, help women structural biologists who are trying to connect with others and get into industry or attend talks. So we have started a LinkedIn group that is called We Inspire Bio. It's a group of women structural biologists, as I mentioned, and anyone who's in the audience who'd like to connect there, either just to sort of join the community or specifically look for connections and mentorship for maybe this transition. I encourage you to go over there and take a look. But in terms of when you're in industry, I think from the point of being a mentee, something I didn't really realize is the importance of choosing your manager, because they're going to be your direct mentor. It's as important as having a great PI during your PhD. And I've been really, really fortunate to join a group where I have a really fantastic mentor who sort of tries to highlight the work of the people in his group, and also helps us sort of grow as structural biologists and as scientists in general. In terms of opportunities to mentor, they are a little bit more scarce than in academia, but we still have students, interns, co-ops that come through. And, in those cases, I think it's a beneficial sort of relationship for both the students that come in that can sort of see what working in industry is like, and then also for the company, because you're getting an opportunity to sort of invest in the future scientists that might be joining your company in the future and might be contributing to the science in the future. So those have, the small amounts that I've been able to do that, it's been really rewarding. And I would encourage anyone who's interested in going into industry that's early in their career, think about doing an internship or just seeing how you like working at a company.
Excellent. Thank you. Thank you, Katie. Roberto, Simone, anything you wanted to add to that?
Yeah, I guess a follow-up question for Katie. Katie, do you think it's possible to sort of have a career in industry where you're in a situation and your mentor and manager are not the same person?
Absolutely. Yeah, that is a very good point. I didn't mention that, but I do. And I guess sometimes you can have formal mentorship that sometimes companies, especially large ones, will have formal mentorship opportunities. But you can also, just through connecting with other scientists at your company, and they don't necessarily have to be in your particular field. But I have some really fantastic colleagues who I do see as my mentors and have given me advice, all the way from the get-go. And again, I think it's being open to having relationships with, others, connecting with them, and learning from them. So yeah, I think that's a really good point to bring up.
Thank you. I appreciate that answer. I think for me, one of the kind of like the early learning experiences was learning to differentiate what is a manager and what is a mentor. And they could be both great assets in your network, but sometimes they're different.
Simone, anything to add?
Yeah, I was wondering if, Kate, you can imagine to have one mentor in academia and one manager in industry, if this would work.
So you mean having still a connection to academia while you're in industry? Absolutely. I mean, I have still kept in contact with my mentors from academia, and enjoy catching up with them or asking them for advice. At times when I'm either struggling or just need some advice and input. So I think especially in today's times, I think keeping connections between academia and industry, really alive, and encouraging them is important. We're all doing science at the end of the day, so we can learn from each other.
Great. And Katie, do you mind mentioning, we have a question that came in. Do you mind mentioning the name of the group once again?
Sure. It's We Inspire Bio. And if you search for that online or on LinkedIn, you should find the group. But also feel free to contact me and I can connect you through.
Excellent. And, while we're here, I just want to invite all of our attendees to please submit questions for the panelists as you like. Just press the Q&A button, which should be at the bottom of your screens. So thanks, Katie, for sharing that again.
Sure.
Why don't we pivot for a minute, if we can, to perhaps Roberto, the perspective from, say, a biotech. How does the approach to cryo-EM differ when you compare it for a younger biotech organization as opposed to a large pharma organizations? What do you think are the unique opportunities and challenges that come with working in a biotech, more of a startup?
Yeah, I think there's some unique opportunities. If I could give myself advice 10 years later, what advice would I give myself? Maybe for the scientists in the room that are looking to introduce Cryo-EM into their companies, I boil down these 4 bullet points, but panelists, please add or refine my answers. The first, I would say, is you want buy-in from your scientific leaders and stakeholders on the value of structure-based drug design. You want a body of people that are going to celebrate that cryo-EM structure when you get it. the best position to be in is when people are dying for that structure, and understand how to use that information to develop develop therapies. The second is focus on projects where cryo-EM is going to have the biggest impact. I drank the Kool-Aid. I love structure-based drug design, but it is possible to make drugs without structure. Hot take. People have done it. So if you want to showcase the power of this technology, you really want to think about like, where is cryo-EM going to have the biggest impact on the portfolio, and then focus on those problems. And then chart out a path to success. What does success look like? And make sure you have multiple threads to success. You don't want to rely on this one experiment to sort of showcase the technology, right? You want to have multiple shots on goal to showcase what cryo-EM can do. I think the third point is, I think this is more kind of on the industry level, is scalability. As you're aligning resources, you want to create a platform that can scale up or scale down with project needs. You don't want to create something that cannot scale down and is very costly, or on the other side, create something that's can handle the capacity and is always struggling to keep up with like the sort of the timelines of drug discovery. Fourth is, it's never too early if you're in a position where you're thinking about doing cryo-EM, but on the fence, it's never too early to start scoping out and having conversations with collaborators. I think some of the follow-up questions that I received are, what are the advantages of, once you've built this pipeline, what are the advantages of processing data in-house versus through a CRO? I think both are options, right? I will say the advantage of processing data in-house is that you have real-time feedback on the data. And as a structural biologist driving the project, one has access to outside data that can guide the data processing. So for example, again, from a small molecule background, are you looking for a ligand that's only partially there versus all completely saturating sample? So again, these are all sort of, biochemical and biophysical data points that can guide the processing, and totally possible to do with, a collaborator, which are awesome, but it just involves communicating a lot more data, internal data to like external collaborations. And that may be more difficult, especially if you're working on something that's like in lead optimization or about to be a drug candidate.
How do you see cloud technology playing a role, Roberto, in processing data in-house?
Yeah, so when I started the Cryo-EM platform at Relay, we kind of had this debate: do we buy a bunch of servers or do we store data in the cloud? Ultimately, we went with the cloud because, again, it gave us the ability to scale quickly up and down. It also has this kind of nice way of tiering data. I think data storage becomes an issue. And you have to start thinking about, like, at some point, is it more costly to store the data or delete the data? And if you need it again, just rerun the experiment. And so I think that cloud computing allows you to sort of store massive amounts of data for cheaper. I'm very nervous about deleting data. And then I think, it's changing. When we started the Cryo-EM platform, spot instances were accessible. I think since a lot of AI technology has come on board, there's a lot more competition for these spot instances. So curious to hear other users' experience. If spot instances are keeping up with the demand of all GPU users, not just Cryo-EM.
Katie, you were nodding your head. It sounded like you wanted to chime in on that.
Well, I guess for the cloud computing, it's I feel like you're spot on with the fact of, I think we're all pretty nervous about deleting data, and it is one of the major issues and one of the major costs. I think that, there are benefits to both. And I think what we've done, at least at Sanofi, is that we have a hybrid system. And so far that has worked really well for us. There are definitely, like you mentioned, there's some challenges for us, and again, another difference between academia and industry. In academia, for instance, CryoSpark instances, as many GPUs as you have is what you can use. Whereas in industry, we are sort of limited by the number of licenses that we have available. And so we can only use so many GPUs at one time. But I think altogether, the cloud computing has definitely been very powerful in order to enable not just industry, but also different academic settings that might not have access to, their own computing clusters to do EM.
Thank you, Katie. So there's a question for Roberto. Is there any cost-effective way for data storage for an academic institute? And perhaps Roberto and Simona, you can both chime in on that.
Ooh. Simona, I'm gonna let you take this one because I don't have a lot of insight into like data storage policies in academia.
I can tell everyone what we do at the FMI. So we basically keep everything, almost everything that goes on papers. So for academics, this could be your criteria. If you have a publication, you keep the data. If you don't publish, you can get rid of it. But the raw data we eventually store on tape. So we decided to go on tape. Which at the moment is fine, but also the data on tape is growing tremendously every year. So in the long term, there might be other solutions that we can consider. At the moment, we store everything on tape for long storage. Seems to be the cheapest solution locally.
I think maybe the only thing I can add to that is just kind of within an academic environment, educating users on data storage policies. I think it varies from lab to lab, and some PIs, they're very busy and kind of just the data storage balloons out. I think it's a good skill to learn early in academia, just to kind of implement a data storage policy and then have all the users comply, right? And I mean, I was one of those postdocs that you write, like I kept my directories really messy, but it's not the best way to do things, especially now these days where we have to be more conscious of like resources.
I think if I could just chime in really quick, I agree with all of those points. And I think something that we can do with having more powerful systems, better, infrastructure. And I think if you have a good relationship with your IT and digital team, developing ways that, people know, as long as your raw data is still there, it might take you some extra time to process it, but you're still okay. So deleting those intermediate files is fine so that people are less nervous about doing that, and having automated ways or very simple ways to transfer data to either cold storage or much cheaper storage facilities. So having those tools available. Also maybe automated checks that can tell you, you're reaching 50 terabytes now, like it's time for you to check, look through your data. Those sorts of systems, I think, will help make things a bit easier for the users as well.
There's an interesting question that came in the chat. Maybe we can take that one, and then there are some questions about skills, and we'll move to that afterwards. But one of the questions asked is, will academia offload a lot of its work or all of its work to industry? Would love to get your perspectives on that. Seems like a loaded question.
I hope not. And I think, speaking off the top of my head, I feel like, you know, at the end of the day, we're all doing science, but academic research should persist and be preserved. And it's driven by different priorities. And I think that priorities between academia and industry are very different. Neither is bad nor worse. But transferring, I think, the workload from academia to industry, I think, would change academic research. I don't know if that's making sense. I appreciate the question, though.
Yeah.
I also don't know whether, like, was the question about offloading to industry in terms of the services? So having, just in the same way as we can like ask for plasmids to be generated now pretty quickly, is that how we will offload like or academics might offload the need for a structure? I think right now still the whole pipeline is way too expensive for that to happen. It's much cheaper to do it yourself in academia than to, use CROs for cryo-EM. Whether the question of science, will academia go away and it will just be industrial science? I completely agree with Roberta that would be an incredible loss to the whole scientific community, because exactly of that fact, the priorities are different. The questions that you have the capacity to ask in academia and spend time on are different than in industry. And academic science fuels industry in a lot of ways and helps us advance. Like, industry, I think we should also say that industry does do basic research in the sense that when we are drugging a target, we're not just looking for the best binder and optimizing the compound. We also want to understand the biology, and that's not always available in academic literature. And we make better drugs if we understand how they're acting in the cell, what's happening alongside, the main target protein. But it's true that this is really a synergistic sort of relationship. And I hope that we just make ties sort of more strong between academia and industry, as opposed to just relying on, or just switching completely to industrial science.
Simone, did you want to add anything?
I agree with everything said so far. I think we should rather foster the collaboration rather thinking to go one way or the other. I think, collaboration is really something that both academia and industry will benefit if it's done in a proper way. So there's lots of method development that academia can do just because industry won't invest time for this. And there are academic labs that we never had the infrastructure to do what industry can do. So at the end, it's a win-win situation if they go on the same path, I think.
Great. So we have a question from Cindy Schwartz. What is your advice for folks who are later into their careers, 20 plus years, in academia or government, to transition to industry? Does education level matter, or is it a combination of education and experience? For example, an early career postdoc versus extensive experience plus an MS or a BS.
Maybe I can start. I would just preface this with saying that I have not hired anyone in industry. So I'm potentially the wrong person to talk about this, but at least from the impressions that I've gotten from talking to colleagues is that there are some differences in the way, and this actually maybe connects back to what we were talking about in terms of the value, or what is the value, or what kind of science is done in different environments. And I think the key difference in terms of the science is that when you are in academia, it's, the main thing is to get that paper done, and you're sort of driving yourself forwards, and what you do impacts you, maybe the lab, but it's a little bit more narrow. Whereas when you are in industry, you're part of an ecosystem. And so what you do impacts the team. And so there are, working in that sort of environment is a little bit different. And I think that if you're a good scientist and you're good at communicating and connecting with people, then I think everyone has the capability of learning that very quickly. So even if you've been in academia for many years, I think you can learn that. There are, of course, other things about, the drug discovery pipeline that you have to learn, and it takes years and experience with different projects to learn. But I think that the transition is not impossible. I do think that sometimes it can be quite hard for people to make that jump later in their career if they've spent a long time in academia to come into industry, but it's not impossible. And your skills, like the skills that you've developed in academia, are very valuable. The knowledge base of the biology, of the techniques, and all of that together, I think is also valuable to industry. So I wonder how maybe Roberto thinks about this in terms of, maybe a biotech versus pharma situation.
I think I agree. I think the skills that you learn in academia are extremely valuable to industry. And it doesn't, regardless of whether you're, fresh out of a training or position like a postdoc or graduate student, or one's been in academia for a very long time. I think speaking about technical skills and just scientific skills, those are very valuable skills. I think it's more of a, having been a few times on the other end, on the hiring end, and looking at someone coming in with a extensive academic background, it's never is like, oh, is this person a good scientist? The science is clearly there. It's more of a, can this person work in the industry environment and be able to be more collaborative and understand that priorities are sometimes driven by a team versus kind of what Katie said, one person driving the priorities to reach the, that publication. So it's more of about the environment, and I think less about like, what technical skills you have. There's no question, like academics, training gives you great technical skills.
Simone, I didn't know if you wanted to add to that at all.
Yeah, maybe something that is worth mentioning is that my advice is when you're finishing your PhD and you want to jump in industry, my advice would probably be go to meetings, try to go to meetings where academia and industry meet, to conferences, and try to reach out to people in front of your poster, try to engage. That's starts on top of discussion and show your interest in what the company does. Yeah, I think that's create your own network and also make your life easier to decide what you want to do in the future just by talking what people is doing. So that's would be my extra advice. Because when you receive, when you're hiring and you receive so many CVs and you look at the science, they are good. They're all good. They're all equally good. They all have a PhD. So, and you don't have the time to question their PhD, right? You trust that's the PhD you got. They all did Cryo-EM, for instance. They all comes for good labs. But then it's really the personality that matters. How the candidate shows interest in what you do. I think that's what I would look for first.
That's good advice, Simone. I agree. It's if you're looking to transition from academia to industry at whatever position level you're at, get comfortable networking. It's it takes a lot of just kind of meeting people and going on a lot of coffee dates, but building up that like initial network is valuable.
Hannah asked a question along the same lines, Hannah Shea, she would love some advice, not just about pivoting from academia to industry, but also about your experience pivoting within your field and within your skills as you develop them. And I think there's a lot there around the networking. It comes back to what we said earlier too about having mentors to help in that. Any other perspectives for Hannah?
I can start this off. It's funny. So Katie earlier asked what happens when the manager and mentor are different. I think it happened for me where someone in my group who was a structural biologist wanted to pivot to something very different, project management. And so this was a case where the person reporting to me had me as a manager, and I was a mentor to going into project management. And so I'll say quickly, I think pivoting to different fields is probably easier in academia. My experience, it's not impossible, but harder in an academic setting. For this case, we kind of, I was very supportive. I strongly believe people should do what makes them happy. And so in this case, we just kind of started a path towards how do we get you to where you want to be, and did like an 80-20 rule where you're like 80% still doing structural biology, 20%, maybe you could be a project manager for a structural biology program, like managing CROs. I won't go into all the details of that, but ultimately that person did sort of like pivot. When it comes to, I think, picking up new technical skills, I think from my sort of biotech-y background, I think it's easy to do in a biotech setting. Everyone has to wear many hats. There's like very loose organizational boundaries. So for me, it was kind of easy to pick up some biophysics skills because like our biophysics team was very busy. So I think it just kind of looking for those opportunities and seeing when there's a need and kind of stepping in.
I agree with Roberto, and I would like maybe just on what you were mentioning about mentoring, I think is interesting. And I think sometimes we, going through, and I've heard sort of a sentiment, at least people that were interested in, going into industry that were still in academia are a little frustrated about the lack of mentorship from their direct, let's say, supervisors or from the academic environment to help them transition. But I think that we can't necessarily expect them to be like that mentor for you because they haven't worked in industry. So there's only so much, help that you can get from that person. So I think that, being willing to, like you said, sort of look out and see if you can find a mentor or even just, one person with experience. I think you'll be really surprised when you connect with a person in an area, then they can connect you and the thing can snowball and you can quickly have really, a nice network of people and learn from a lot of people. I think transitioning to doing different skill sets, we're still, for example, I started doing I went the reverse to what Roberto had. So I started with, diving into EM, and now recently I also added crystallography to my skill set. And so that was a bit of a pivot, but because the need was there and, I had good support from my mentors, it was really a fantastic experience. And I think as scientists, we all love to keep learning, and gathering these new skills is an opportunity to do that. So if you have the opportunity or the drive, then go for it, connect to people and grab those opportunities if you can.
Okay. So, staying on the conversation about skills, there's a few questions about, what are the technical skills that you see being most relevant for going forward, and what are the softwares that people are tending to use, and there's actually quite a few questions about tomography and how that plays a role going forward. I'll invite any of our panelists to chime in on that.
Maybe I can start here.
Yeah.
Start from the first part of the question. So what skills you need? That's a very difficult question, because also the structural biology field itself is kind of evolving, right? So what we are doing today, probably in 2 years, is going to be quite different. And so it's hard to suggest what kind of skills you have to learn. For sure, you need to be familiar with IT in our field, in structural biology in general. So having a good background in computational I think is essential. But I also think it can be learned later on. So yeah, this would be probably quite important to have as a main skill. Cryo-EM image processing for single particles is becoming more and more friendly, user-friendly. To want to make, I mean, if I can mention the name of the software, but then I mention all of them. CryoSpark and Relion, probably the most popular, and these are the two software that you must know if you want to do cryo. There is no other way around, I would say. Also there is lots of automation going on, so the learning process is also faster than a few years ago. Yeah, that would be probably the skills. And for the second part about tomography, that's of course a hot topic at the moment. In the academic, in industry, there are discussions whether to go this way or not, and it's still, I think, at the early stage. I wish I will see tomography soon as a technology in the companies. I'm for it, but one has to look at the projects that are available now in the industry. I mean, we should go to a kind of change of what are the expectations for tomography in terms of, at least for industry, which are quite different than if you think the expectation for tomography for academics. So yeah, I hope to see tomography in the industry soon, but so far I have not seen flying everywhere yet, but I hope this will change soon.
Thanks, Mark. Katie, do you want to go?
Yeah, I generally agree. I think especially when talking about skills for sort of, the next generation coming into cryo-EM, I think that we're at a time where it's maybe getting harder and harder to predict, because with the computational capabilities that are developing at a really rapid pace at the moment, I think it's hard for us to predict exactly how those will integrate, how they will work and change our work. So while for now, I completely agree that, being able to use the command line and maybe script some things and generally be good at that kind of stuff is really, really useful. I think just as we saw, CryoSpark sort of revolutionize a little bit the way that we do data processing. It's much more GUI intensive and much easier for a lot of people to navigate, even if you don't have that experience. So I think that thinking about what skills you want to develop going forwards, being able to do the work that we have to do now, of course, but also thinking about what your value is as a structural biologist in the future. And I think that while, I think most directly, we bring structures that show interactions that tell us about potential compound binding. That's one aspect to it. But in another sense, we also more broadly impact biology, because we understand these machines in terms of their three-dimensional structure. We know that they're interacting in space, that they're malleable, that how changes at one end of the molecule can affect another. And I think that contribution is important for biology. So I think that in terms of skills, maybe the practical kind of hands-on, maybe robotics will take a little bit longer to advance and we will still be needed hands-on at the bench. But then also thinking about maybe data interpretation and integrating your structural insights with biology more generally will be really important. And I think that feeds into tomography, or the question about tomography. And while right now it's still, it's a question in industry, how are we using this, or how is it going to be useful? As the technique advances and the computational capabilities and the insights advance, I am absolutely sure that it will be valuable. And understanding, sort of getting past the scales that we're sort of stuck in, maybe atomic level, but transitioning to the cellular level has been harder. And I think the more that we can now bridge the different resolutions, then we'll learn more and understand biology better. So I'm excited to see what happens in the future, but it is a little bit hard to predict, I think.
Yeah, I appreciate those answers. I think speaking strictly about technical skills, I think having some ability to understand scripts and write scripts, it will always be valuable, even if like me, if you're like a bench lab scientist. Again, what we do involve, it's a multifaceted and cross-collaborative discipline. So learning how to, talk to IT and communicate needs is valuable. But I think, to Katie's point, as a structural biologist, the field is evolving with all the new advances. It's, delivering structural information on an unprecedented scale is cool, but more, what does that all mean, right? Figuring out biology and mechanism of action. I think that's where I think this real structural biology happens. Figuring out, we're studying these things outside of their native context, and how do we connect what we're doing in a test tube or in a Cryo-EM grid? How does that going to lead to like a therapeutic outcome, or isolating the target in a way that's relevant for drug discovery? I think those are sort of for me, I feel those are the heart of the questions that we deal with in structural biology. So I think those building on those skills and just thought processes that help answer those questions is the most valuable thing I think we can do.
Excellent, thank you. Well, the questions are pouring in, so we'll keep them coming. Josh White was asking, in contrast to the previous question about academia offloading on industry, do you think that closer ties between academia and industry will be driven by the general availability of high-end microscopes in academic institutions? And looking ahead, how do you see Cryo-EM evolving at national centers, core facilities, individual research labs, at industry with respect to education, development of new technologies, and fees for services? So a lot in that question.
Yeah, maybe I could just say that it's already happening. So, in industry, of course, we try to have our own, we have our own facilities, but sometimes we do need to outsource data collection, and we do work with universities that have available time, and that works really well. I think so far that's been a real benefit, especially, if you have one microscope and it goes down, for example, you need to fill the time. We have to keep going. I think the pace is sort of fast in terms of how we need to generate data. So being able to utilize time at universities is really valuable for us. Maybe I'll let Simone and Roberto take the rest of the questions about the national centers.
My hope would be that like cryo-EM sort of evolves and this follows the same trajectory as crystallography, where in crystallography we saw consortiums, companies essentially buy a beamline at a synchrotron. And so my hope is that that sort of continues in making cryo-EM more accessible to all users. I think there is a trend, even companies co-owning an instrument, which is difficult. Because of all the sort of the legal framework in that, but I think it's totally possible, or even cost sharing, right? There's some models where like company buys a microscope, but an academic houses it and they take over like the maintenance of the instrument. So I think there's so many ways between academia and industry to work together and just make this acceptable.
Yeah, I agree. I think national centers are essentials for, especially for medium-sized biotech, which they cannot, either they cannot afford a full EM facility, or because they have projects that maybe have a lifetime of 2-3 years, and they don't know if Cryo-EM will be a tool that we use for many years, or it's just a need for now, and that's it. So there's this to consider. So that's why national centers definitely make sense, and I'm happy they exist. For big pharma, I have a different opinion. I think big pharma must have their own EM facility. This is my thought. When you have so many projects, you want to have a fast feedback or whatever you put on the microscope, and at the end, it's not much more expensive than NMR spectrometer. So I think, yeah, big pharma must have their own EM facility. That's my opinion, and I've seen how this works so well for Novartis, and this seems to be working very well also for other big pharma. And no doubt, EM will contribute to drug discovery and structural biology for many more years. So it's a good investment.
I think just to add to that, I think that's an important point, and why sometimes we think about it's very useful to have CROs and other opportunities for that. Oops, I'm sorry.
Simone, maybe I can follow up with that while Katie comes back. I think it's very valuable when large pharma buys microscope, because that also sends like the right message to a smaller biotech. Smaller biotech always sort of tends to follow what large pharma is doing, and so it makes the buy-in for biotech easier when we see like the large pharma industry partners going in on cryo-EM. So it affects everyone. When large pharma buys microscopes, it has rippling effects.
Yeah. Katie, what were you gonna chime in with?
Oh, sorry. Yes, I was going to say that, if you're able to do the science in-house and have that, there are sometimes insights that you get from the data while you're processing it, while you're looking at it, while you're looking at your sample, that, will help sort of figure out things for the rest of the project. And sometimes I think those can be lost when you outsource or when someone else processes your data, or, So I think in that sense, there is really a value of, like you said, having it on site and being able to not only quickly turn around, but also see how things are behaving and maybe get some insights that, you weren't even looking for in the first place.
Thank you. So we're just about up on the hour, but if I could ask one question to each of you, when you look at the future of cryo-EM, what is the emerging trend or technology that particularly excites you? Did we stump our panelists? So I don't know if anybody wants to chime in on that.
I guess I'll chime in. I didn't talk about tomography, but I'm excited to see how tomography sort of plays out in drug discovery. Is it when I think of drug discovery, there's different modalities, gene editing, biologics, small molecule. I think TBD where tomography comes in, but I'm super excited to see hopefully play out in all three areas or, yeah.
I think what I'm excited about is our capability to, or growing capabilities to learn more from the data that we have. So in the processing sense, like, there's so much that we, currently I think are still missing. We have these potential to see more about the dynamics of our system, learn more about the movements of the molecules, and learn what's important in terms of their mechanistic function. And then I think that advancements in automation in the processing, and then also in model building, and then understanding all data in more in a wider sense with other, predicting their structures, predicting interactions, alongside the experimental data. I think it will be really exciting to see what happens with that.
And I can add that I would be excited with any technology that would enable to expand the target that we actually have. So there's been like this for Cryo-EM. Cryo-EM really changed the field in industry in terms of drug discovery. It enabled many more targets that you could not think to solve before. GPCR one obvious examples, but there are other many targets that are now possible to have three-dimensional structure that was not possible before. And in the future, yeah, for sure, tomography, as I said before, I wish this will contribute to drug discovery. And also all this new protein design, there are new tools coming every month. And that this has been shown already to be very useful to design binders and soft structure that are not possible otherwise. So that's also a field that I'm quite excited about, protein design and yeah.
Excellent. Well, thank you very much, Roberto, Simone, Katie. It's been a pleasure, and on behalf of all of our attendees, on behalf of Clovertex, we're grateful that you agreed to be panelists for our discussion today. Mazdak, thank you for your help in organizing this whole webinar. And as you pointed out, there will be more coming in this series, so please keep an eye out for that. To all of our attendees, we appreciate your questions and your participation. And I want to go ahead and close the webinar and wish everyone a wonderful rest of their day. Thank you.